So inspired in data from early clinical trials with TIL therapy for the treatment of patients with metastatic melanoma, different investigators have proposed different approaches that could be used to target cancer. On one side, a lot of discussion was around the generation of more defined T-cell products, mainly using TCRs that target specific target antigens that are specifically expressed on cancer cells. There's been some data presented about how not only TCRs with high affinity are important for tumor or antigen clearance, but also TCRs with lower affinity. And in fact, one of the investigators proposed to generate a T-cell product that has diverse and it's polyclonal in the sense that it recognizes multiple targets and also multiple TCRs for each target with different affinity. And he believes that this may provide better protection long term. From Chris Klebanov's group, we heard a little bit about TCRs that have been isolated that could be used for therapeutic TCR gene-engineered T-cell therapies targeting a specific driver mutation in NRAS, well, 3 different driver mutations in NRAS. His group has isolated TCRs targeting these targets and has shown potential for treatment in preclinical models. And I believe there's more data to come in the near future regarding the efficacy of these TCRs in the clinic. There's been another short talk by Reno Debets where he presented a new target, a new potential target to develop gene-engineered T-cells targeting using TCRs. This is a target called Roporin-1. It seems to belong to the family of cancer germline antigens. And in fact, he describes already TCRs that could be used therapeutically in the future. In the case of my own talk, in fact, I summarized some of the clinical experience with TIL and presented several different projects that under our point of view in our group, we are investigating to improve T-cell therapies. One intends to enhance and enrich the tumor-reactive T-cells, the neoantigen-reactive T-cells, for enhanced efficacy. Another alternative is to exploit and leverage the tumor-reactive T-cells that are in peripheral blood through the isolation of specific subsets of T-cells in the circulation that are enriched in these reactivities, and also the isolation of novel TCRs that target different hotspot driver mutations that are recurrent mutations that can be that are frequently mutated in cancer and could help develop novel therapies.